Students, in the previous video we were studying the ecological external adaptations in the hydrophyte with respect to their habitat and in which environmental conditions they grow. Now we also has to learn the internal adaptations in the different organs of the halophytes. Different organs in the sense we have to see the internal adaptations in their root, in their stem and also in their leaves. What are the internal adaptations are developed in the halophyte? There are two kinds of roots are seen in the halophytes, most of the halophytes. Some roots are underground, they are subterranean roots, and some roots are aerial roots, they are called as nematodes. The subterranean roots are, or all the roots are of stilt root or prop root kind. They are produced for giving the more support, mechanical support to the halophyte. So we will see the internal adaptations in the subterranean roots root of the halophytes. So for studying the internal adaptations, we have to take a transverse section of subterranean root. And then we have to observe under microscope and we have to observe the different anatomical layers or differentiations from epidermis to the center. Though the root is very young, then also it shows the very thick cuticles or many layers of the cuticles even at the young age of the root. From very you will see that externally the root has a several layers of quark, and these quark layers are developed from the quark cambium. There are many layers are developed where because most of the layers are get peeled off and the new layers are get added by the quark cambium cells, and that's why we can see the several layers quarks are developed at the periphery of a root. Then the cortex of a root is also of outer cortex and inner cortex, several layers cortex we can see. But the special features of cortex are the cortex in case of halophyte roots is made by the star shape or stellate cells. That cells have the lateral arms, and with the help of that lateral arms, the cortex cells are connected with each other, and that's why we can see the different pattern of arrangement of a cortex cells in the halophytes. These cortex cells have a special kind of thickening along their ridges, and this thickening is lignified. So each cortex cell arms are heavily thickened, and that is of a lignin kind. That thickening is observed from the lignin compound, and that also gives the better mechanical strength to the root. Some of these cells of cortex are filled with oil and tannin. You will also observe the many cells of cortex are found filled with oil and tannin intermittently. Then the fifth cell at the center, the fifth cell of the root are also found thick wall with a pickled kind of thickening, and they also contain the lignin and tannin. They are lignified and they also contain the tannin and oil. All these features you can see, most of the features are support for the mechanical strength by the deposition of oil, tannin and lignin, and they are heavily thick cells are present in the root, and that also for supporting the mechanical strength to the hydrophytic plant as well as root. Now we will see the internal ecological adaptations in the stem of hydrophyte. The stem of hydrophyte is also very soft. That is also when you will take a transverse section, you will observe that the epidermis cells are heavily cutinized, even though the stem is at a young stage. The outside epidermis you will see or you will find the thick cuticle present The cuticle is present because they can do the two functions. They provide mechanical strength and also they can check the rate of transpiration. The epidermis is also heavily thickened. The cells of epidermis are spherics and they are also found filled with the oil and tannin. Next to cuticle, the epidermis layer is there. The squarish type of cells are present. That are also heavily thickened and found filled with oil and tannin. Then next to epidermis, you will see the hypodermis. The hypodermis is also multi-layer or several layers thick. Their walls are also thick. Thick wall cells are present in the hypodermis. Then next to hypodermis, there is a primary cortex divided into the outer cortex and inner cortex. This primary cortex is also multi-layered or many layered. This cortex possesses the thickened cell filled with tannin and oil. So here actually in the cortex cell, tannin and oil deposition you will find. Some of the cortex cells may also possess the calcium oxalate crystals. In between, some of the cells shows presence of calcium oxalate crystals. In between, you can also see S-shaped heavily thickened spicules present in cortex, and these heavily thickened S-shaped spicules are present to provide a mechanical strength to the cortex as well as to the stem. The inner cortex possesses the branched thick-walled cell called as a scleroid. So in between the inner cortex cells, you will see the scleroid cells that are also nothing but a deposition of calcium, and they are of star shape, and they can also perform the function of mechanical strength. Then next to cortex, there is a presence of multi-layer sclerocamentous pericycle. So pericycle, that is surrounding to the stele or vascular tissue. Normally you see single layer, but here that is a multi-layer and also sclerocamentous in nature. So that sclerocamentous nature says that the pericycle can also perform a function of mechanical strength for the stem. In the center, pith is present. That pith is also multi-cells. And that pith also shows the presence of S-shaped spicules. These spicules also contain tannin, and the reason behind presence of these spicules is nothing but a support to the mechanical strength. In the stem, you will see the vascular tissue are well developed. The vascular tissue well developed for performing a better conduction and transportation of the food material. These are the somewhat ecological adaptations in the root and stem of halophyte. You can also see, for details, you can also see the figures or TS of root and TS of stem, and you can observe the anatomical structures with the ecological adaptations in the halophytes.
This figure is a TS of halophyte root. From outside you can see there is a multi-layer cork produced by cambium. That cork shows the presence of lenticels. After that there is a secondary cortex, then primary cortex made by stellate cells. Stellate cells arranged or join one another. Then there is a pith at center. So you can refer this figure. This is a figure of TS of stem. Shows thick cuticle, epidermis, multilayer hypodermis, then outer cortex with a S-shaped spicules, air spaces called lacuna, and oil and tannin cells are there. Inner cortex shows star-shaped scleroids. Endodermis is there. Multilayer pericycle we can see. The vascular tissue also seen well developed. Outside there is a phloem. Inner side there is a xylem. Xylem is also very well developed, and at the center there is a pith, parenchymatous, with empty spaces called lacuna, tannin cells and oil cells, and also shows the presence of spicule. This is what a ecological adaptations or the internal structure of halophyte stem. You also has to refer this for the detailed study about the anatomical adaptations in the halophyte stem.
Now we will see the internal adaptations in the leaf of halophytes. The leaf of halophyte is dorsiventral, they remain green, they remain always green. Outside, when you will take a vertical transverse section of leaf, you will find the upper epidermis and lower epidermis. The cuticle is very well developed on the upper as well as on the lower epidermis. Then the epidermal or epidermis cells are also heavily thick, and they may also contain calcium oxalate crystals. So this modification is for checking the rate of transpiration and for giving a mechanical strength to the particular organ of a halophyte, especially for leaf. In the leaf, the stomata is of sunken in nature, that is present in the cavity, and the stomata are confined only on the lower epidermis. This is also a reason for to check a rate of transpiration, because the lower surface of the leaf is not get easily exposed to the sunlight, and that way the stomata is remain closed most of the time and stop the loss of water. The reason is that though the halophytes are present in water, but physiologically they are present in a dry condition, and that's why they show the more adaptations that are of xerophilus in nature. Then you can see below upper epidermis there are several thin walled cells are remain present that are called as hypodermal cells, and most of these such cells are filled with the water, and the outer side cells are seen filled with the oil and tannin. Means the inner hypodermal cells are full of with the water, and the outer hypodermal cells are seen filled with the oil and tannin. The water filled cells provide water, and oil and tannin cells provide the mechanical strength. Then below this hypodermal cell, the lowermost layer of this hypodermal cell are mucilaginous and get embedded in the palisade tissue. After hypodermal cells, you will see that mesophyll cells are well differentiated into a palisade and spongy tissue. The lower layer of hypodermal cells is mucilaginous, and that get embedded in the palisade tissue. As I said, mesophills are well developed and they are well differentiated. You can also see quirky areas present on the ventral surface of a leaf. Quirky area that is on the lower surface of the leaf. So these are what we several ecological adaptations we can see in the leaf of halophytes. For more details, we have there is a need to observe the figures of this section with respect to root, stem, and leaf. Then you will see what are the ecological adaptations or what are the differences in anatomical structure as compared with a normal plant and these plant organs.
This is a section of vertical transverse section of leaf. We can see upper and lower epidermis covered by a cuticle. Then towards upper side there is a subepidermal aqueous cells that stores water, also stores oil, tannin, and some of them are becomes a mucilage cells that get embedded in the palisade tissue. Mesophyll cells are well developed and well differentiated into palisade and spongy, Palisades are seen rich in chloroplast. Their function is photosynthesis. And spongy are spherical. Their function is storage. Then you can see the lower epidermis only shows the presence of sunken stomata that are also seen covered by a cuticle, and single layer hypodermis is seen below the lower epidermis, not at the upper epidermis. This is what a anatomical structure of halophyte leaves. You can see the adaptations like aqueous cells, mucilage cells, then sunken stomata, thick cuticle on the above, upper and lower surface, lower epidermis, and stomata confined only to the lower epidermis. So you also has to refer this figure for adaptations in the leaves.